Podemos.
Okay. Good morning, Steve. It's a pleasure to have you here participating in this International Symposium of Postgraduate at the State University of Maranhão, UEMA. UEMA thanks you for accepting the invitation to give a lecture on anaplasmosis in humans and animals, biology, ecology, diagnostic, clinical presentation, and treatment. We will present a brief curriculum vitae of our speaker first, and after, feel free to present your talk. John Stephen Dumler is professor and chairperson of the conjoined departments of pathology at the Uniformed Service University of the Healt Sciences, Waterreed Nation Military Medical Center, and the Joint Pathology Center. He has a long-standing interest in tick-borne infections, especially focused on tick-borne rickettsial infection. He has published over 270 peer-reviewed scientific papers and authored more than 110 chapters in textbook of medicine, infection diseases, and microbiology. The majority of his research is conducted centering on pathogenesis and molecular mechanisms by which Anaplasma phagocytophilum causes diseases. Other specific research interests include vector and tick-borne diseases and pathogenic agents, especially obligate intracellular bacteria in the other Rickettsiosis. Success in your talk.
It's a delight to be here to talk with all of you. I'm very pleased that I have the opportunity to speak with my Brazilian friends once again for so many years. May we begin the talk, please?
Yes.
So, can you see the presentation?
You can see.
Okay. I have a few disclosures to make about funding and my relationship with the United States military and Department of Defense. These are my opinions and don't necessarily represent those of the university, the Department of Defense, United States Army, Navy, or Air Force. I'd like to start the talk by illustrating how I became involved. I was working as a postdoctoral fellow in the laboratory of David Walker, who was a famous American rickettsiologist. And he received a blood smear after writing a paper on a new disease in America called Ehrlichiosis. And the blood smear came from this man, Johann Bakken. Sorry, I have to go back. Dr. Bakken was an infectious disease physician who lived in a part of the country in the United States where ehrlichiosis did not exist. And he described a man who was 81 years old who was hospitalized in June of 1990 after 4 days of fever, headache, confusion, muscle aches, and weaknesses. He had been bitten by a brown tick 10 days before admission to the hospital. On a blood smear, bacterial clusters, as shown here, were observed in circulating neutrophils, and were interpreted to represent a Staphylococcus infection. Unfortunately, the patient did very poorly and died within several days, and at post-mortem examination, we were able to identify these same structures inside of neutrophils in multiple tissues from the patient, this from the spleen. This suggested to Dr. Bakken that this might be a form of ehrlichiosis that had not previously been identified in the United States. Between the 1990 and 1993, 11 additional cases were identified by Dr. Bakken in the upper Midwest part of the United States, in Wisconsin and Minnesota, and identified to be related to an Ehrlichia species, but likely to be a new species to infect humans, although well known to veterinarians in the United States and in Europe. Over several years, we were able to not only identify these cases, but with the help of another colleague, Jesse Goodman at the University of Minnesota, we were able to cultivate the organism from horses as well as human blood in 1996. And we're able to study this from a molecular standpoint. Here you can see the clusters of organisms inside of cultured cells from humans. These are HL60 cells. This was a new tick-borne disease in the United States that we subsequently characterized as being related to Anaplasma Marginality and called Anaplasma phagocytophilum. Over the years, tick-borne diseases have increased in their incidence and prevalence in the United States and around the world. The data that I show here are derived from the United States Centers for Disease Control Morbidity and Mortality weekly reports and accrued over the years 2011 to 2019 at a time when tick-borne diseases were rapidly increasing in the United States. The total data represents 365,000 cases of tick-borne disease reported. Most of these were Lyme disease, but among a large group, you can see many of these are rickettsial organisms, such as spotted fever rickettsiosis, ehrlichiosis, anaplasmosis, and other related organisms. Over this period of time, Lyme disease was the largest concern in the United States. And over this period of time, the number of cases and the incidence of Lyme disease in the United States increased approximately twofold. However, if one examines the data for the other diseases at the same time, you can see that the highest increase in case numbers and incidence over this period of time were not related to Lyme disease, but were related to human granulocytic anaplasmosis, the disease that Dr. Bakken discovered in 1990 in Minnesota. A little bit of a background on rickettsiosis are owed to the man who discovered the first rickettsial organism and after whom the genus, the order, and even species are named, Howard Taylor Ricketts. Dr. Ricketts was assigned to study Rocky Mountain spotted fever in the western part of the United States in the early 1900s and discovered an organism that was transmitted by ticks that could cause fatal disease in humans. Subsequently, it was realized that other organisms were the causes of many rickettsial diseases. Historically, epidemic typhus, which is caused by Rickettsia prowazekii, and the disease known as louse-borne typhus, might have had a role in the outcome of the Peloponnesian War, clearly had a role in the European exploration of the Americas, Napoleon's defeat in Russia, and in influencing the outcome of World War I, where disease was the main determinant of injury and death, as opposed to actually injury from traumatic processes on the battlefield. As I said before, Rocky Mountain spotted fever was discovered by Dr. Ricketts in western part of the United States and has subsequently been shown to occur broadly across North America. And in Brazil, this is known as Brazilian spotted fever, caused by the same organism. The organisms that now comprise rickettsial infections are considered to be very broad and include those that infect endothelial cells, that include members of the genus Rickettsia and Orientia, those that include Ehrlichia and Anaplasma that infect macrophages and neutrophils and other professional phagocytic cells. Classically, Coxiella burnetii, that infects predominantly macrophages, has been considered in this because it is an obligate intracellular bacteria. And Bartonella have also been considered Rickettsia for some time, although phylogenetic activities will show you that these organisms are distinct from the classic Rickettsia at the top of this slide. The order Rickettsiales now consists of a number of different families. The major families that cause disease include those in the Rickettsiaceae, that include Rickettsia genus and Orientia genus, and separately the family Anaplasmataceae, for which the major genera are Ehrlichia and Anaplasma. All of these organisms are alpha proteobacteria, and they have small subunit RNA genes that are often used to characterize their relationships by genetics. These organisms are also clearly bacteria. They contain DNA, RNA, ribosomes. They divide by binary fission, as you can see in the image on the lower right. They have a gram-negative type of the cell wall. And unique to them is that they have their life cycle that is within an arthropod host at some point. The organisms Bartonella, or formerly Rochelainea, and Coxiella, although considered Rickettsias, actually are not within the Rickettssiales order. This very difficult to read slide is simply meant to show the extent of the order Rickettssiales now with the molecular generation of data. That includes the Holosporacie, mitochondria. These are the mitochondria within most eukaryotic cells that are closely related to the Rickettssiaceae, to the Anaplasmataceae, and another group of organisms called the Midichloriaceae. A higher magnification of a similar analysis based upon the coding frames of these shows similar relationships, with the Anaplasmataceae very closely related to the Rickettssiaceae that are down here. All of these organisms have reduced genomes. That is, they contain many genes that do not encode for any protein, and they become dysfunctional over time because they are not required for their life inside of other cells. A few examples of the organisms that I would like to talk about today include Ehrlichia chaffiensis, the original cause of human ehrlichiosis in the United States; Anaplasma phagocytophilum, the organism that Dr. Bakken discovered in humans in the United States, that had been known by veterinarians for many years before; Anaplasma marginality, that infects erythrocytes, perhaps the most important form of anaplasmosis in the world. In addition, other species include those in the genus Neoricketsia, such as Neoricketsia ristici, which causes Potomac horse fever; Rickettsia rickettsii, that was discovered by Dr. Ricketts, the cause of Rocky Mountain spotted fever or Brazilian spotted fever; an interesting intracellular bacteria called Wolbachia, in this case Wolbachia pipientis, that actually provide important properties of life to insect cells as well as to some helminths. The current classification of Anaplasmataceae within the Rickettssiales actually has about 6 unique genera, some of which are candidatus because the organisms have not been cultivated. I'd like to focus my attention today on the organisms in the Anaplasma genus that you see here. What's interesting is that because of the genomic revolution, there are now more than 1,500 genomes from Rickettssiales organisms, including 4 from Midichloriaceae, 11 Holosporaceae, 167 from Rickettssiaceae, and 1,340 from the Anaplasmataceae, of which 1,250 of these are Wolbachia genomes. So we will know very much about the genomes and perhaps deeper about the biology of these organisms as time passes. I'd like to focus now on the human diseases, and then toward the end, I will discuss a little bit more closely how the human diseases inform us about the animal diseases and vice versa. In the United States, there are several forms of ehrlichiosis, or human anaplasmatacea infections. The first is human monocytic ehrlichiosis, which is caused by the organism Ehrlichia chaffiensis. And it gets its name because the organisms grow inside of a peripheral blood cell called a monocyte, as you can see in this image. The disease that Dr. Bakken discovered in humans, caused by Anaplasma phagocytophilum, the disease known as human granulocytic anaplasmosis, gets its name because the organism grows inside of granulocytes in the peripheral blood of humans and infected animals. There are other forms of ehrlichioses that affect humans, including Ewing ehrlichiosis, caused by Ehrlichia ewingi, which is genetically similar to Ehrlichia chaffiensis, but it grows in neutrophils and is phenotypically similar to Anaplasma phagocytophilum. New entries include Ehrlichia murus, identified in the upper Midwest United States, the Panola Mountain Ehrlichia, which is similar genetically to Ehrlichia ruminantium, that has been identified in the southeast part of the United States, Ehrlichia canis, a worldwide distributed organism that causes disease predominantly in canids, has been identified as a human infection in some parts of South America, chiefly in Venezuela. Neoehrlichia micurensis has been identified as a severe disease, caused thrombosis and severe infections in Europe, and a mild infection in Asia, particularly in China. And another form of anaplasma, Anaplasma capra, identified as a cause of febrile illness in China in humans, and probably maintained in ruminant cycles with ticks in Asia. Regardless of which form of ehrlichiosis one thinks of here, all of them present as febrile illnesses that may not have other differentiating features. Sometimes the laboratory findings can be very helpful if one is trying to make a diagnosis should treatment be required. The question that is asked at this point is, how often do these diseases occur? Again, these are data that are taken from the Centers for Disease Control in the United States, morbidity and mortality weekly reports, accrued from 1986 through 2019. And one can easily see that over this time, there has been a linear increase in the total numbers of these cases. And the vast majority of these tend to be caused by Anaplasma phagocytophilum or human granulocytic anaplasmosis. Here's the map of the incidence of human granulocytic anaplasmosis in the United States. It was first discovered in the upper Midwest, where I'm pointing now, but subsequently, by 2006, was tabulated to be in these regions as well as in the east and the northeast parts of the United States. By 2008, the numbers of cases were increasing and the distribution around these areas was expanding. The same in 2010, 2012, 2014, where the cases are larger in number. So this disease in humans, caused by Anaplasma phagocytophilum, has an average annual incidence between 2014 and 2018 of about 12 cases per million across the United States, but considerably higher in specific locations where the disease occurs more often. The incidence increases as age increases, which is very unusual for tick-borne disease, and it's the highest in individuals between the ages of 50 and 70 years. And there are more men infected than women generally. About 6% of people that were reported to be sick had an immune compromising condition of some sort. 1/3 of all people that were diagnosed were hospitalized, and 6% were considered to have a life-threatening complication. 7% of these individuals required admission for care in an intensive care unit. The overall case fatality rate is not high, 0.3%, but it's considerably higher in those that are over 70 years old. So the elderly are more at risk. The organism is transmitted by Ixodes species ticks, typically nymphs, but also by the adult stage ticks that I've illustrated over here in their unique individual geographic distributions that are also depicted on the map. The reservoir seems to be generally small mammals, perhaps deer, and perhaps ruminants, and it could well be that the species that are described or the strains that are described in each one of these small mammal or large mammal reservoirs has unique features, and it's believed that some of these do not have the capacity to cause disease in humans, while others do. How often do humans become infected around the world has been an important question. And so there have been many groups around the world that have done seroprevalence studies in human populations. Overall, one can see that the median rate of seroprevalence is relatively high in most places: Asia, Europe, North America, generally between 4 and about 10% of all people screened have antibodies indicating that they were previously infected. If one looks at studies that focus only on cross-sectional populations, these numbers are a little bit lower because they include many asymptomatic people who have never had the disease. But if one begins to examine populations that are at risk, farmers, people that work in occupations that take them into the woods where they would be exposed to ticks and other conditions like this, you can see that the prevalence here becomes higher in each of these situations in all locations. And if one looks at other populations known to have a tick-borne disease, especially an Ixodes transmitted disease like Lyme disease, one can see that the rates of these are even higher. The seroprevalence rates are higher for studies in Europe and North America. So this is clearly associated with tick and tick bites. Estimates of how often these diseases occur in the United States have been made based upon passive recording of cases by the Centers for Disease Control and are as high as 80 cases per 100,000 per million in Wisconsin or Connecticut, two endemic regions in the United States. But active surveillance shows that there are many more cases detected by this method than by passive surveillance in both these locations, for a ratio of active to passive surveillance of an average of 11-fold more. Therefore, the estimates of disease prevalence based upon passive surveillance are significant underestimations of the actual numbers of cases that occur through these methods. The seroprevalence based upon these data over these years would be reflected by the numbers on the right panel here. The disease itself, as I said before, is characterized largely by fever and perhaps headache and other muscle aches, as one can see here. And you can see how this compares with other tick-borne diseases, such as Lyme disease or Rocky Mountain spotted fever, with the chief difference being the absence of a skin rash as compared with Rocky Mountain spotted fever or Brazilian spotted fever, and compared to other diseases that may occur and present similarly. It would be very challenging for any clinician to be able to distinguish this disease from any of these others in a single patient because of the undifferentiated nature of the clinical presentation. The laboratory findings can be helpful, however, in that most people with this disease, human granulocytic anaplasmosis, have some degree of low white blood cell count and presence of immature band neutrophils in the blood smear, as well as thrombocytopenia, low platelet counts, and to include mild evidence to moderate evidence of hepatic injury as based upon increases in alanine transaminase or aspartame transaminases in the serum. Among those individuals who have central nervous system manifestations, unlike Lyme disease or Rocky Mountain spotted fever, these are rarely seen with human granulocytic anaplasmosis. A diagnosis can be made by looking at a blood smear, such as through a Wright, a Giemsa stain, or a typical malaria smear, or perhaps with greater sensitivity in a Buffy code analysis. But the sensitivity is variable between 25 up to 75%. PCR is a very good technology for diagnosis with a sensitivity between 90 and 95% during the acute phase of illness, and reactivity that diminishes rapidly after treatment. There does not appear to be a chronic phase in humans, but there is a brief persistence of DNA that can be obtained from blood after the beginning of therapy. Culture, which does work, is generally not available and has an unknown sensitivity and typically is not used for diagnosis. Perhaps the most important technology for diagnosis has been the use of serologic testing. The problem with serologic testing is that most people are tested at the acute phase of illness, and while some may actually have antibodies at that point, they probably reflect pre-existing infection. And most people that actually have active infections have a seroconversion such that 99% are seropositive at 1 month after their acute phase sample has been taken. And 70% of those individuals show a seroconversion or have a 4-fold increase in their antibody titer. One of the problems with serology for Anaplasma phagocytophilum is that antibodies can cross-react with other closely related species, such as Ehrlichia chafiensis. The sensitivity of IgG testing is greater than 82%. And as is true for most rickettsial infections, IgM sensitivity is poor and specificity is low. IgM is not advocated for use for diagnosis of rickettsial infections. The current treatment for human disease is dependent upon adult or children and the disease, but generally involves the use of a tetracycline antibiotic, mostly doxycycline. The dosages are listed on the right side of the panel. Tetracycline itself can be effective for both adults and for children. This is not contraindicated in children. And an alternative would be the use of rifampin in adults or children, although this has not been well tested. None of these drugs has been tested in randomized or placebo-controlled studies. Currently, there are no vaccines available for humans, although a vaccine for Anaplasma Marginality exists for bovines. It can be prevented by performing regular tick checks on people and on pets, but it must be noted that transmission of Anaplasma phagocytophilum can occur within 24 hours of a tick attachment, but is more efficient when the tick is attached for 24 to 48 hours. This implies that the sooner the tick is removed, the more likely transmission is to be blocked. Tick repellents can be used when outdoor, including DEET, permethrins on the clothing, or a variety of other new tick repellents that are effective alternatives to DEET. And these are registered by the U.S. Environmental Protection Agency and can be accessed at this website. Other advice would be to protect your pets from tick bites by using appropriate repellents, collars, or other methods. Limit exposure to tick-infested habitats. Wear protective clothing when you're outdoors, such as long sleeve shirts, pants, socks, closed-toed shoes. And remove attached ticks rapidly. I don't advocate the use of folk remedies such as gasoline, kerosene, petroleum jelly, fingernail polish, or to try to burn a tick from the skin. Often causes more harm than it causes good. Among individuals that have become infected and were reported, complications include hospitalization of 30%, death in 0.3 to 0.6%, life-threatening complications in 6%, but also include things like renal failure, acute respiratory distress syndrome, meningitis and encephalitis, pneumonia, disseminated intravascular coagulation, and sepsis. So the disease can be quite severe. Here are some severe and rare complications of Anaplasma phagocytophilum infections. I won't read these, but you'll have this to know that these are a variety of inflammatory conditions that can occur in the presence of Anaplasma phagocytophilum infection in humans. I wanted to talk just briefly about some of the research and biology. And one of the things that we've been studying has been, how does Anaplasma, when it infects a neutrophil, affect the function of Anaplasma by reprogramming its genome and its transcription? Of course, neutrophils, once they encounter a microbe, often go through a process of activating specific functions, including degranulation, inflammation, motility of the cell, and the production of proteases, chemokines, and chemotactic factors that favor inflammation. And this happens with Anaplasma when it gains access to the cell. However, other factors that often happen with exposure to other microbes are deactivated by Anaplasma, including respiratory burst, apoptosis, endothelial cell adhesion, transmigration through endothelial cells, phagocytosis, and microbial killing. The net effect of this is that Anaplasma reprograms neutrophils so that they survive longer than uninfected cells because of the delay in apoptosis, and this allows them to grow to a larger number of bacteria that will actually induce inflammation and attract other neutrophils that will allow the expansion of the bacteria by infecting new hosts. These cells that are in the blood that are infected do not marginate or attach or emigrate through vascular barriers, and this increases the overall concentration of infected cells in the blood that will be accessible when a tick feeds. It also increases the likelihood of inflammatory injury in those individuals who are infected. We studied this by examining humans to find out about inflammatory cytokines and noted that humans infected with Anaplasma in the acute phase have high levels of interferon gamma and interleukin 10, a pattern also replicated when we developed a mouse model of the disease, as you can see in the right panel. Of interest, we wanted to study what would happen in this mouse model. And here's the pathology of the liver for a wild-type mouse. And one can see a small amount of hepatitis with these small lesions in the liver parenchyma from this infected mouse. If one were to remove interferon gamma, which promotes these inflammatory lesions, one can see very little inflammation. Interestingly, if one removes interleukin 10 in an interleukin 10 knockout mice, interleukin 10 controlling interferon gamma, suddenly the inflammation becomes much more exaggerated and very dangerous. So the balance between interferon gamma and interleukin 10 will determine the inflammatory outcome to some degree. This is interesting because the more interferon gamma is present, the more inflammation there is present, as one can see here. However, the interferon gamma does little to control the pathogen burden. As you can see, there's little inflammation here, but very large number of pathogens. So this brings up the concept that Anaplasma actually causes severe disease because of the cytokine hyperproduction and the induction of a systemic inflammatory response. And these things are often characterized by many of the features that we see with anaplasmosis. Some other conditions like this include sepsis, acute respiratory distress syndrome, which we see, macrophage activation and hemophagocytic syndromes, which typically require only a moderate to low pathogen burden, and they're driven by a number of genetic abnormalities, or it can be acquired. Typically, it is actually driven by the presence of cytotoxic lymphocytes that lead to expansion, and under normal circumstances, these cells are controlled by the cytotoxic lymphocytes. In hemophagocytic syndromes or macrophage activation syndromes, the feedback here is lost because infected cells are not killed, leading to a cyclical process of pro-inflammatory development. In humans, we studied this by looking at cytokines and compared them with control patients. And what one can see here, based upon the p-values, is that there are a number of pro-inflammatory cytokines that are part of the macrophage activation syndrome that are elevated with anaplasma infection. And these relate to severity. The higher these are, the more severe the disease is. We studied this in a horse model. The horse also becomes infected and diseased with Anaplasma phagocytophilum. And to suppress this inflammatory process at the level of the transcription factors STAT1 and STAT3, we treated horses at the time they became ill with dexamethasone. And in each case, it's easy to see that the clinical disease is less severe if one suppresses the inflammation, but there are more bacteria when one suppresses it. So the disease is really caused by inflammation that are driven by the bacteria, but the bacteria alone themselves cause little pathologic injury. I'll go rapidly through this. This is simply to demonstrate some in vivo work in mice that we've done that shows that cytotoxic lymphocytes are not functional with anaplasma infection. And as a result of that, we believe that this may be related to the inability to control long-term infections in some animal species. So the immunopathogenesis of severe human granulocytic anaplasmosis is dependent upon the organism that triggers inflammation and innate immunity. Disease severity is related to the expansion of these cytotoxic lymphocytes that produce interferon gamma. The immune and inflammatory responses are modulated by transcriptional changes that seem to be mediated by infection of that particular cell. And the severity of the disease in humans, horses, and mice, and perhaps others, relates to the lack of the ability of these cytotoxic lymphocytes to function properly and to control the infection and the inflammatory disease. Severity can be actually modulated by altering these inflammatory cytokines. Anaplasma impairs cytotoxic lymphocyte function, and this possibly leads to the loss of critical regulatory pro-inflammatory control of cytokines and induction of cytokine release syndrome, such as macrophage activation syndrome. So the most important points about human disease are that it's not rare and can be severe. You can prevent this based upon the use of early empiric treatment and clinical suspicion, which includes identifying tick bite, an unrevealing physical examination, a patient with fever, leukopenia, thrombocytopenia, and elevations in hepatic transaminases. And diagnosis can sometimes be confirmed rapidly with blood smear or PCR testing. Serology of acute phase blood is not appropriate for diagnosis, but comparing acute and convalescent serology is. The outcomes of the disease are generally benign, and it depends upon early treatment with doxycycline. Chronic or persistent human infection is not proven, and co-infections occur in less than 10% of the patients. A role for prophylactic antibiotics has not really been investigated. No vaccines are available, but the disease can be prevented by avoiding tick bites and other methods. Now, I've talked for a long time about human disease, but I noted earlier that perhaps the most important disease that's caused by anaplasma are not humans at all. They are bovines and others. And this diagram shows that particular distribution. And when one can see, this is the only place in the entirety that humans seem to be greatly impacted. So this was taken from a recent publication on which I was the senior author. That's the citation there. Looking at some of the biological, ecologic, and epidemiologic features of anaplasma species. This is a very large group, but I wanted to really focus on two of these species for comparison: Anaplasma marginality and Anaplasma phagocytophilum. And here you can see that both of these diseases can cause significant disease in animals, not only in humans, where Anaplasma marginality has not been demonstrated to cause any disease. So bovine anaplasmosis, caused by Anaplasma marginality, is characterized as a disease that has fever and increasing bacteremia over 3 to 7 weeks after infection. The disease itself is characterized by a significant drop in the packed cell volume of anemia. And this is generally believed to be caused by phagocytosis of infected erythrocytes. In addition, one sees icterus, lethargy, weight loss, abortion, and death. And animals remain persistently infected for their life, and they become reservoirs for tick transmission to naive cattle, an important disease worldwide. Tick-Borne fever, which is generally the name that's given to the disease in bovines caused by Anaplasma phagocytophilum, mostly occurs in small ruminants in Europe, probably mostly in sheep, but can occur in goats, cattle, horses, and dogs. In sheep, it can lead to a high fever of many weeks duration, leukopenia of both granulocytes and lymphocytes, and secondary infections that seem to be common to background immune suppression, including Staphylococcus aureus, pasteurellosis, and Listeria monocytogenes sepsis. I want to wrap up by talking about two things that have been understood by study of both Anaplasma marginality and Anaplasma phagocytophilum, and I think have shed a lot of light, especially in view of the COVID-19 outbreak that we're seeing globally now. One thing that is known about Anaplasma marginality is that it accrues to very large numbers in the blood. So it's characterized by persistent bacteremia. This bacteremia is permitted because the organism has antigenic variation, so that during the first period of time, the population increases, immunity begins to mount, antibodies remove the bacteria that express one antigenic variant. The remainder of the population begins to grow, and antibodies to develop to that population and eventually control it, but not fully. And the reason for that is a good question. A similar thing seems to happen to a smaller degree with Anaplasma phagocytophilum, where antibodies will control subpopulations as the organism goes through antigenic variation. However, an interesting phenomenon that has been seen in Anaplasma marginality infection and may be present with Anaplasma phagocytophilum, hasn't been studied well, is the presence of T cell exhaustion. This is related to this high, long-term, persistent bacteremia that one sees. And it's depicted here by the presence of T cell function over a long interval of time after they've been immunized. Over time, T cell function fails in all these circumstances. And they fail because of a process by which long-term persistent exposure to high numbers of bacteria or viruses can actually lead to changes in expression of T cells so that their function is dampened, a process called T cell exhaustion. This seems to happen in many virus infections. We're beginning to have evidence of this now with coronavirus infection. And I believe this may be true not only with Anaplasma marginality, but maybe the explanation for the immune suppression that we see with Anaplasma phagocytophilum in sheep, and perhaps the severe sepsis-like syndromes, the cytokine release syndromes we see with Anaplasma phagocytophilum in humans. So with that, I think I've probably exceeded my time. I'd like to stop and thank you for listening. And I will open up the podium again to the moderator so that I can entertain any questions you might have. Thank you again.
Thank you. And I loved your talk about Anaplasma phagocytophilus because you tried to find in animals here in Brazil, but no success. Exists one Anaplasma, but we don't know what species. And we tried to talk with you about this and how to solve this problem, but I and Marcos have this problem, and we don't know how to solve this. But I have some questions to you about the circulation of morulus, similar to Anaplasma phagocytophilus in neutrophiles in dogs and cats in Brazil, if you know about something.
I do know. I know of some of these reports, and of course, I've talked with Brazilian scientists about these. The morphology is not highly specific, and we can see similar morphology in neutrophils because of infections and because of other biological processes. So it becomes important to be able to do a laboratory test that will provide more definitive information about the nature of the organism. Typically, this would be a PCR assay to look for one or more genes that would confirm exactly what that microorganism is. I know I've seen this conducted in Brazilian cats, and I think probably there are some cats that sustain this infection. I'm a little concerned about the data about the extensive cat infection that has been published out of some of the Brazilian studies. But I think that this is the important way to be able to do this carefully, is to use carefully controlled, well-established laboratory methods to identify what the infectious agent is.
If people want to do some question, please, we have time to do the answers. Steve, you believe that the problem in Brazil maybe is about the tick species, because in USA, the Anaplasma phagocytophilus is transmitted by exodotes. And the species of ticks in your country, they do not have in Brazil. You believe the problem maybe is or not?
It's likely to be part of the problem. I think the more we examine the species in the genus Anaplasma, the greater diversity that we find. For example, there are many scientists that believe that the genus Anaplasma should be split, and that those erythrocytic anaplasmas like Anaplasma Marginality, Anaplasma Centrale, Anaplasma ovis, and Anaplasma capra probably should be defined as a unique genus separately from Anaplasma phagocytophilum. And the reason for that is there are many unique genes and probably many unique biological functions. And I think the more we examine these organisms, the more we examine their vectors, and the more we examine their reservoir hosts, the more likely we are to find reasons why such distinctions exist. From my standpoint as a physician, I'm very interested in why organisms cause disease. But for many of these anaplasma, they seem to be commensal organisms that do not cause significant disease in their reservoir hosts. It's only when they inadvertently are passaged through the vector into a non-definitive host, like a human or a horse or a dog or another animal that they're not accustomed to being introduced into, that disease can occur. The alternative for that I think is beginning to become evident is that there are genetic predispositions among all populations, whether humans, dogs, cats, or wild animal species, for which the polymorphism will either prevent severe disease or prevent any disease or allow severe disease to happen. And understanding these things will be the big challenge in the next 20 years.
Thank you. I have another question. In Maranhão, here in Maranhão, we found 8 positive horses for Anaplasma phagocytophilus, targeting 2 targets for the MSP gene. What other genes would you recommend testing to really confirm the aging like Anaplasma phagocytophilus?
Yes, MSP2 is a very nice target for PCR because there are typically many copies of this gene in the genome. For the Webster strain, for the HC strain, there's 115 paralogous copies in the genome of Anaplasma phagocytophilus. The problem is that these are all unique per species. And so the use of this actually probably is not the best choice for trying to phylogenetically characterize anaplasma. The ANK-A gene seems to be highly conserved. Its presence is highly conserved among all strains of Anaplasma phagocytophilus. It's likely that it is a critical gene and would be a very good target. There's been good phylogenetic work done with this particular gene to characterize it into specific clades that correspond to certain animals or the reservoirs. And of course, there are a number of other genes that are part of the housekeeping genes, if you will, the genes such as surface genes, such as RNA polymerase genes, or things like that, that are in most bacteria that can be also used in concatenation with many other genes to be able to establish an identity. Truthfully, we really do need to be thinking more about genome sequencing, and the technologies are becoming available to be able to do this now by doing next generation sequencing, taking very small fragments of DNA and using ultra sensitive methods to be able to reassemble partial genomes and compare them on a scale that we couldn't do until recently. That would be my advice.
Okay. Another question. If you believe that the vaccine using Anaplasma Central will be provide immunity, partial cross immunity to Anaplasma Marginality, it is a good alternative to control anaplasmosis?
Well, it's a very good question, for which I'll state right away that I do not know the answer to this. But the reason that Anaplasma centrale was developed was because it seems to be genetically very closely related to Anaplasma phagocytophilus, and the disease process is considerably milder. And I believe that there's data that illustrate that it has some protective efficacy. I don't know that it actually can completely eradicate the organism, but understanding its biology will allow investigators to identify those key items that will allow that organism to become a better immunogen to protect it. The problem is, as I referred to in the very last slide, is the process of T-cell exhaustion. And I really think this is a very fertile area for future investigation. In the United States and in Brazil and other places, in humans, we've been studying this process in individuals with cancer. Why don't cancer vaccines work? They don't work because the antigens that are presented to the T cell are always present, and they induce the T cells into a dysfunctional state, T cell exhaustion. The same process is happening in anaplasma in cattle. If one can actually use processes to reactivate those cells, to remove the signals that deactivate them, I believe that you can see much better immunologic control with another vaccine candidate, whether it's a live vaccine challenge or whether or not it's a subunit vaccine. That, I think, will be the future to understand that process better.
Lisa Young say, Steve, can you give your contact for us?
Of course.
Yes, I can send. No problem. Lisa, please, could you send me email and I can send you the contact of Steve. Oh, no more questions. I would like to thank you. And we are so glad to see you online. And the presentation, the better presentation. And I hope that you can present another talks for us in the future.
Thank you very much. I'm very happy to do so, and I hope to see everyone in person in the near future.
Okay. Bye-bye.
Please stay safe. Bye.
Bye.